So we got the sound that we need. All right, Dr. Matthew Chahati, PhD in bioinformatics. I got to say that slowly because I heard it last night. Of course, I've read it, but it's a field that I had not heard of before I became familiar with Matthew. But bioinformatics, which has to do with mathematical models, I suppose, related to the biological fields.
Like DNA sequence analysis, protein analysis, like databases.
DNA sequence analysis, protein analysis. Okay, all right. Those sorts of things. Bachelor of Science in Software Development, that's kind of your area, at least computer science. From the University of Szeged, is that correct? In Hungary. Okay, MA in Religion from Greenville Presbyterian Theological Seminary. So he is not fluent in Greek and Hebrew, but he's conversant in the biblical languages, active creation science researcher. This is all on our website, but we don't always read some of the things that are posted and available to us. Active researcher in the field of creation science since 2007, has written 45 creation science papers. Speaker for CMI Creation Ministries International from 2019 to 2020, worked at several academic positions now in California, doing, tell me again?
It's a
pharmaceutical bioinformatics research company working particularly on the pandemic and the virus, the coronavirus, or not?
We're studying cancer.
Cancer genes. Okay, excellent. And he has worked in the assembly of the Neanderthal and Denisovian genome sequences, active in molecular baraminology research. If you heard his talk yesterday, he was defining baraminology. And if you're first checking in, some of you probably know this already. Most of you probably do. Bara, the Hebrew verb to create. Min is the Hebrew verb for kind. So the study of created kinds, what that means. And without any further ado, I will turn it back over to Dr. Chahati.
I apologize, folks. This is my fault. Things,
Let's go. PowerPoint saved to station. Best way to do it. Then I'll bring it back to PowerPoint.
Okay, so greetings everybody, and welcome back to the Society of Creation conference. And so this will be the second part in a two-part series on baraminology. In the previous presentation, I talked about morphological baraminology, also known as classical baraminology. That's how this science started. And so I'll be talking about the basics of molecular baraminology, which is a field which I'm very active in. I've written a lot of papers in this newer sub-branch of baraminology. And for just a few words about the science itself, for those who are just joining, the verse in Genesis, actually several verses, verses 12 and I think maybe verses 12 and one of the other verses, they talk about how God creates different animals and plants that multiply each according to their kind. So the Genesis text doesn't mention a binomial nomenclature, rather it mentions reproductive communities, that it doesn't talk about different species, but rather about different kinds of animals or organisms which each reproduce according to their kind. So we shouldn't be forcing our modern view of taxonomy onto the biblical text. Rather, we should perform exegesis and see what the text really says about species and about organisms. So last night we talked about morphological baraminology. Now I'll talk about molecular baraminology. Also, I'd like to make a few comments about some things that we said last night about science and about religion. Creationism is science. Science did not begin with Darwin. We creationists, we study the handiwork of God, not the hand of God. And also, just because science studies nature only doesn't mean that there's nature only. Doesn't exclude the existence of God. Just because the focus of science is a natural world, it doesn't mean that the supernatural realm doesn't exist. Who is to deny that science cannot be done, or can be done in a supernaturally created world? According to the philosophy of science, science itself doesn't deal with the reason why things came into existence. There can be supernatural reason for the existence of the world. And so if nature was created supernaturally, then we can still do science in a supernaturally created world. Also, science has its origins in Christian Europe in the Middle Ages, but also in Genesis chapter 1, verse 28. This is the biblical mandate for science. Then God blessed them, and God said to them, "Be fruitful and multiply, fill the earth and subdue it. Have dominion over the fish of the sea, over the birds of the air, and over every living thing that moves on the earth." So the key here is that Adam must subdue the earth. And in order to do so, he must know and understand it. This is science. So God is telling Adam, be a scientist. What did Adam do? He became a taxonomist, and he named the animals. So science has its roots in the very first chapter of the Bible in Genesis. We creationists, we can be proud of this. Also, just a few words, because it's been in the news. Roe versus Wade has been overturned. God is good. God is love. Praise God. God created us in his image. God created life. God created all things very good, without pain, suffering, disease, or death. And there's a great, Roe versus Wade overturned means that more babies will be saved. As a human species, there's a big variety of human diversity. Also, there's a great diversity of biological life, which we shall now examine from a molecular perspective. Now, last time we left with this picture here, and I asked you, the audience, do you think that these three animals are they one kind or many kinds? One's a fish, shark, reptile, ichthyosaur, and then we have a mammal, which is a dolphin. And so morphological studies could classify these three animals into the same kind, into one big kind. If you just look at superficial morphological similarities, even though they are different classes of vertebrates. We also learned yesterday that the level of the kind is around the family. And so the question is, how would we classify these organisms based on their DNA? So maybe the DNA is the key to understanding how we can better classify these three animals, not into the same kind, but into separate kinds. So why molecular baraminology? What is the impetus? What is the reason for this separate sub-branch of baraminology? Looks may deceive, as we saw previously. Two or three organisms from different groups may look alike, like the shark or the dolphin. So we can't separate them based on physical traits. Rather, let's look at their DNA. We can tell the difference based on their DNA. As we saw in Nathaniel Jeanson's talk, there's a wide variety within a Brahmin. For example, in bovids, there's a big variety of morphological similarity. So again, let's look at their DNA. Maybe we can see some very sharper boundaries between kinds based on their DNA. Also, there are missing morphological characters, incomplete skeletons, only cranial characteristics. There's a lot of incomplete morphological data. What about microorganisms? We can't measure the cranial capacity of a bacterium. So let's look at their DNA. And I have done several studies when I looked at archaea or bacteria or viruses, looking at their DNA sequences. And also, it's important that your DNA determines your physical traits. Your genes determine proteins which carry out functions in your cells. That is, the genotype defines the phenotype. The genotype being your genetic makeup, and your phenotype being your physical makeup. So our genes determine how we look, what we look like. So molecules before morphology is an important principle here in molecular baraminology. So what kind of data is out there that we can use for molecular baraminology studies? Now, bioinformatics is a very rapidly developing science, and that's an understatement. And I would highly encourage young Christian college students, if you're studying biology, get into bioinformatics. There's a huge ton of data out there. It's lying there, hasn't been analyzed yet. You can get your fingers really dirty in analyzing this ton of data. Billions of sequences in the public domain, all waiting to be analyzed by thousands of algorithms. The data is also accumulating faster than we can even analyze it. It's a big, huge field. As creationists, we have to harness bioinformatics. Scientists are sequencing larger quantities of DNA with increasing speed and decreasing cost. The Human Genome Project was $1 billion on the range of several billion dollars, but now you can get yourself sequenced for $1,000. That's how much the prices have dropped. This is really radical stuff. So as creation scientists, it's a question of how we should make use of this data, not whether we should make use of the data. We need to make use of the data or get left behind. We really have to get entrenched into bioinformatics. So just to show that this is a growth of GeneBank, which is a major bioinformatics database. Here's the year 2000. And so we have data for 2008. You can see the exponential growth. It's a literal explosion of data. We have to make use of the data. This is 2021 statistics. We have 215 million protein sequences. We have data for 115,000 organisms, lots of data out there. There's a number of genomes sequenced over time. And it's only for 2014. Again, we see this exponential curve. In 2022, there's even more genomes sequenced. I have to keep on stressing that there's a ton of data out there. We have to lay our hands on it. So now let's talk about terminology and methods. The goals and terminology of molecular baraminology are the same as morphological baraminology. And for those of you who are just listening now, a baramin is a kind. It's a biblical kind. It's the term that we're going to use all the time to define a group of organisms that can interbreed with one another, but which do not interbreed with other organisms. Like cats are one kind, dogs are one kind, horses, there's the human kind. These are all kinds. They intermix with one another, but they don't interbreed with other organisms. Molecular baraminology uses different data, different techniques, different technologies based on molecular biology. There's the gene content comparison, the whole genome sequence analysis. These are algorithms that I myself have developed. And we also do mitochondrial and chloroplast genome sequence analysis. And I'll describe these things in detail as we go in the presentation. Here are just some summary papers. If you want to read more about these individual algorithms, maybe you can take a snapshot. They've been published in several creation journals. Also, at least I think a couple of these were stealth publications, which were published in secular journals. Thoroughly scientific. And so what is the goal of molecular baraminology? There's a threefold goal: to glorify God by cultivating science that honors him. 1 Corinthians 10:31 says, therefore, whether you eat or drink or whatever you do, do all to the glory of God. To think God's thoughts after him. We have a scientific program in the Bible. We want to discover the kinds of organisms that God has created in the living world. Only God knows what kinds there are, but as scientists, we also want to discover what these kinds are. We need to think God's thoughts after him. We also want to show that science speaks about the glory of God and not just random evolutionary chance, that we are intelligently, wonderfully created and designed. And also to show that the Bible has answers. As Christians, we can do science as well as evolutionists. We're even better because we have the truth. I believe in this. I believe in this. We also want to point people to Christ revealed in the scripture, that the scripture is actually and factually true. Not just about Genesis, but also about how you live your life. Also about Christ. And so therefore we can share the gospel with other people. So what kind of molecular data are there in the databases? There's a whole genome sequences from the very first DNA letter to the last. The human genome is 3 billion letters. genome of a bacteria is about maybe 1 million 1 million DNA letters. There are gene-protein sequences that we can analyze, we can compare with one another. There's mitochondrial and plastid DNA whole genome sequences from these cell organelles, which we can align with one another and cross-compare between species. Databases, there's NCBI, which has whole genome sequences, organelle genome sequences, gene sequences. UniProt is a protein database. There's a Galaxy server where you can compare protein content between two species. And I'll talk about in more depth about different methods of molecular baraminology. One is the whole genome K-mer signature method. It's a method where we analyze the DNA signature of these DNA words, which are distributed in the whole genome sequences of different species. There's the gene content method. We compare what percent of genes do two species have in common. If they're in the same Brahmin, they have a lot of genes in common. If not, they have a smaller common gene content. And there's mitochondrial genome sequence analysis, also chloroplast genome analysis. The first algorithm, which I'll talk about, is a whole genome K-mer signature algorithm. Has a long name. So we're studying the whole genome sequence. We're studying these short K-mers. K-mer is a short stretch of DNA. The DNA letters A, C, G, or T. K-mer is K letters long. If you have a tetramer, it's ACGT is a K-mer of length 4. And so the way we have to understand this is that just as a backup, just as an aside, if we look at different literary genre, we have magazines about sports, which contain words such as score, stadium, team, or player. These are words which you would expect to encounter if you read a sports magazine or a book on sports. If you talk about architecture, you'll find words such as building, bridge, engineer, or blueprint. These are vocabulary words which are very characteristic of architecture books. Genetics, we read about base pair, about genomes, chromosomes, and mutations. We don't really expect to read about scores, like soccer scores in a book about genetics. Neither do we expect to read about chromosomes in a book on architecture. So they're literary genres with a very specified professional vocabulary. Now, how does this translate to genetics and this K-mer signature algorithm? The thing is that during creation week, God created different kinds, each with its own created genome, also known as a baranome. Bereshit bara Elohim, vat shamayim vat aretz. There's the word Bereshit bara, which means he created. And so a created genome is a baranome. It's a special technical term that Danish creationist devised. And so a baranome, at the very starting time point of creation, it is pristine, it is unaffected by deleterious effects of mutations, it is pluripotent, it is uncommitted and undifferentiated by mutations. That is, through time, it can accumulate mutations in this genome, and then it can differentiate into the genomes of different species which make up that Brahmin. The genomes of species within a kind originate by mutational differentiation from the baranome, and here's a picture whereby we can understand this process much better. This is a wolf, which is the archaebrahmin of the dog kind. It is the first original species according to Baraminology, that all other dog species came from the wolf. And I think that even secular scientists admit that dogs form a monophyletic group, otherwise known as a Brahmin. And so the first species would be the wolf. Everybody started out from the wolf. And so then we have one branch leading to the coyote, has several mutations in these letters. Then we have the St. Bernard with some more mutations in different positions. Another branch out to the golden retriever, different mutations in different portions of the genome. And so the whole idea is that the archaic dog genome differentiates into the genomes of other dog species in the dog kind. So the idea is that we expect to find a very similar DNA signature in the genomes of all these dog species. There's going to be a very common DNA signature in the genomes of these individual species, which all belong to the same kind. If you look at cats or horses, there's going to be a different signature. We can detect this using statistical methods. But so going on, we'll see that a DNA vocabulary word, a K-mer, is a short DNA segment. For example, here's CCGTGACG. It's 8 letters long. It's an octamer K-mer. And so we can look at the distribution of these octamers in the genomes of these species. These words can be regulatory motives, structural or repeat elements. And so just as I mentioned about architecture and about sports and about genetics, just as with literary genres, species from different kinds each have a different DNA vocabulary. They speak a different genetic language. From different kinds, their word content correlates very poorly from different kinds. But if you have species from the same kind, they have a large overlap in their DNA vocabulary. They speak the same genetic language, very similar vocabulary. Their DNA word content will correlate very well. That's what we expect according to this model. And so indeed, again, this paper is a self-publication published in a secular journal. And so what do we see here? This is a square matrix. That is, it's a visualization of a square matrix with all these similarity values. And the values, they range from -1 to +1, since we're measuring the correlation of the DNA vocabulary between two species. So for example, we see Anopheles, these are mosquitoes, Glossina or tsetse flies, Drosophila or fruit flies. And the reason it's a symmetric square matrix is because if we take, let's say, two Anopheles species, let's say Anopheles epioticus and Anopheles merus, we see that it corresponds to a very bright yellow color. The bright yellow color, the brighter the color, means that it's closer to +1. And in statistics, +1 is a very good correlation. It means that these two Anopheles species, their DNA vocabulary correlates very well with one another. They speak the same genetic language. These two species, they correlate very well, whereas Drosophila ananaceae and let's say Anopheles maculatus is a dark orange. The darker the color means that it's less correlated. And so a Drosophila, a fruit fly, and a mosquito, their DNA vocabulary will correlate very poorly. And this is the way you can differentiate between species of the same kind and different kinds. So here we find a block of Anopheles, Glossina, Drosophila. We see that each square is going to represent an individual kind. We see that species in this kind, their DNA vocabulary correlates very well with one another. We have this bright block, these bright colors here. Whereas if we compare species from different kinds, there's going to be a darker color and it'll be a very low correlation. So in this manner, we can detect individual kinds which are very, very disjunct, very separate from one another in a very statistically significant manner. And this was first published in a secular journal, a first study of its kind. I also studied bats. I mentioned bats yesterday, and somebody asked like how many bat kinds are there, and I studied bats based on this algorithm, also on mitochondrial studies, and so we found that there are 4 bat kinds. And of course, here color schemes may vary. So in this color scheme, the red blocks correspond to higher similarity values. I made these figures using RStudio, and there are these different parameters as to what kind of coloration scheme I can use. The color schemes vary, but when we look at these images, we have to look at like different, we have to look at these blocks here. we have to look at these blocks which correspond to different kinds. And so I also studied mammals, also another Cell publication. And so it's very interesting. In mammals, I studied mustelids, which are like weasels or otters or minks, these kinds of animals. Also compared them to bears and also cats or felids. And so the subject of this study was, I was comparing the red panda, because it's a really interesting question. How do we classify the red panda? Eilurus fulgens is its Latin name. And before the age of molecular studies, different taxonomists said it looks like a bear, it looks like maybe a cat, maybe a raccoon. They weren't really sure as to how to classify this very interesting animal. So with the onset of molecular baraminology, we can look at its whole genome camera signature, we can look at its DNA, and so we can find that this animal, it fits in with these other mustelid species. I would say that it looks very much like the American mink. It's Mustela americana. You can see that there is some sort of similarity: whitish head, red body color, and so according to this algorithm, this animal fits in with different mustelid species. I would say that Eilurus fulgens, the red panda, is a mustelid. It is not a bear. This is Ailuropoda melanloica, which is the giant panda with this orange star. The giant panda fits in with all the other bears in the bear kind. Some other taxonomists, they said it looks like a cat. I also studied cats. It's not a cat. The red panda is indeed, it's a mustelid, according to this analysis. I also studied the whole genome camera signature of a lot of other animals. I found, studied pinnipeds, which is this block right here. We have bears, dogs. We have different, we have 3 whale kinds. Here's the hippopotamus by itself. And lo and behold, we also have bovidae right here. Of course, I did this study independently of Dr. Jeanson's analysis. I did not know he was going to talk about bovids, about the big variety of bovids. As we saw yesterday, that we saw all these bovid species, we were tantalized as to where the boundary is, where the morphological boundary is. So this really illustrates nicely that if we look at the DNA, we can find a sharp distinction between bovids and non-bovids. So we find that here's Capra sibirica, the Siberian ibex, which is a bovid, and also Ovis aries, the sheep. And so we were talking about the sheep buffalo kind, and all these other species. We have buffaloes, we have bison. This is a molecular evidence of the existence of the bovid kind. So independently, morphological studies and also molecular studies come to the same result. And this is really just a wonder of creation science. Then the next big algorithm is the gene content method. Physical traits measure things like body mass, number of fins, presence or absence of a particular bone. Now let's look at how many genes 2 species have in common. Genetic traits are also part of taxonomical descriptions. In taxonomy class at college, you'd basically think that, taxonomists, you look at wings or legs or cranial capacity, but you can also look at like internal organs, or you can also look at DNA characteristics. These are also part of how we describe animals in a taxonomy study. So genes produce proteins which code for different traits. Genes which fulfill the same function in different species are called orthologs. So indeed, we have genes in common with chimpanzees, with horses, with dogs, even with yeast. But of course, the more genes we have in common with the more genes the 2 species have in common, mean that they're more closely related. It means that they're from the same kind. So let's measure the genetic similarity between 2 species based on how many similar genes they have, how many orthology groups they have. Now I'm going to talk about it's a sort of a technical interlude. It's not a problem if you don't understand this. the basic concept is let's look at how many genes two species have in common. But we can measure this by looking at orthology groups. The gene content method assigns each gene or protein in a species proteome, that is, all the proteins in a given organism, to a so-called orthology group. An orthology group is made up of several genes, virtually the same function, from many different species. These genes differ ever so slightly in their sequence, very, very small sequence dissimilarity, but the sequence difference doesn't affect the protein's function. It's just a synonymous mutation. Doesn't affect the function of the protein. For example, there are alcohol dehydrogenases, cyclin Bs, or histone proteins. These, like histones, they occur in all sorts of genomes. They have a function in the structure of the genome. Like flies, frogs, sheep, fish, they all have histone proteins in their genome. They all fulfill the same function for coiling the DNA around themselves. OG identifiers are listed, and so therefore we're comparing the orthology group ID content between two species. So just a picture of what is an orthology group. We have 4 clusters. These are NOX genes. There's NOX1, NOX2, NOX3, NOX4, and Fort Knox. And then we have this new protein. We don't know what it is. We don't know its function. And so we can take this protein sequence and compare it to all these orthology groups. And so we compare to NOX1. Is it a NOX1? NOX2? Knock, who's there? And then there's, and then is it a NOX3? Whatever group it'll be most similar to, it'll be assigned to that orthology group. So we take this protein, and then we'll give it an orthology group identifier. So now this was the technical part. It's really not so important. If you don't really fully understand it, it's not a problem. The main idea is we're measuring the gene content similarity between two species. And so how do we do that? We measure this Jaccard coefficient value. And so if we see on the Venn diagram here, we have two species, species A and species B. We have the intersect of their gene content. So A is the number of genes in species A, B, the number of genes in species B, then A intersect B is the number of common genes, and then A union B is the union of gene content between species A and B. And so here's the equation for the Jaccard coefficient value. It's basically the intersect divided by the union of all the genes in these two species. It's going to have a value between 0 and 1. And of course, if we have two species from the same Raman, we have a high overlap in their gene content, high proportion of similar genes. And so the JCV value is close to 1. These are two species from the same Raman. If we have two species from different Ramans, there's only a very slight overlap. And the two species might even have a very disparate number of genes in their genome. So this is how we tell, based on this model, whether two species belong to the same Raman or to different Ramans, based on this algorithm. So I studied this first in NCLDVs. That's just an acronym. NCLDVs are very interesting organisms, or highly interesting. They are nucleocytoplasmic large DNA viruses, such as these ones. They're not koosh balls. So some people think that they're very degenerated bacteria. Some people think they're viruses. I think that they are actually bacteria, not really viruses, despite their names. As a comparison, here's the rhinovirus. Here's the HIV virus, a very, very large size difference between different viruses. I think that basically because of the size and the size of their genome, they have a 1 megabase per genome, 1 million DNA letters. I think they're really bacteria. HIV has a genome of 10,000 letters, if I'm not mistaken. SARS-CoV-2 has 30,000 DNA letters. And so what I did was, again, we have a heat map or a dark map. And so here we can see that there are 5 groups, 5 clusters with at least 3 species. We have this group here, we have another group, and then we have these five groups here with at least 3 constituent species. So we can say that we've discovered 5 putative NCLDV hologramins. But now I also did this for Archaea, 160 species, and based on this analysis, I found 8 hologramins based on the basic chemical that they metabolize, methane, either sulfur or nitrogen metabolism, or salt ions, holophiles. What this means is that we have these 8 groups here, and they each metabolize either methane, carbon, or sulfur or nitrogen. The idea is that the physiological processes which metabolize methane or sulfur are very, very fundamentally different from one another. So much so that let's say a methane metabolizing archaea cannot just simply transform into a sulfur metabolizing archaea via random mutations. The pathways and the regulatory pathways and the genes that are involved in the process of using methane are very, very, very different from those which use salt ions or nitrogen or sulfur. So archaea, it seems they're different. They differ based on their basic metabolism. So we have these 8 groups in archaea. In insects, we have 104 species. We have 5 groups from Lepidoptera, which are butterflies, Hemiptera, which are bugs, Hymenoptera, which are I think ants and bees, to I think 2 dipterans, which are flies. We have these 5 groups in insects. In primates, we have humans, New World monkeys, and Old World monkeys. And also in these analyses, I also added some outlier groups for comparison. We have Monodophis, which is a possum, mouse, just to show that that these outliers, we should expect to see great differences between outlier species and the species that we're studying. That's the role of outlier species, to demonstrate real true differences which we can compare bound differences to. There's also mitochondrial genome analysis. A mitochondrial DNA, or mtDNA, is a very short DNA sequence, about 16 to 20,000 base pairs. It's easy to isolate. Mitochondria is very abundant in the cells. They're little batteries of the cell. So cells need energy in order to live and thrive. They're also very easy to sequence, and so therefore they've generated a lot of mitochondrial DNA sequences for a lot of organisms, for 24,000 different species at NCBI database. The downside is that the mitochondrial DNA represents only a very small fraction of the entire genome. So you can draw some conclusions from mitochondrial analysis, but you can't really generalize to the whole entire genome. But it's still, easy and useful to analyze mitochondrial genomes. And so what we do is we take the mtDNA sequence from all the species in a study. We do a multiple alignment, and then from the multiple alignment, we can calculate the sequence similarity between any 2 given sequences / species. So for example, so again, this is the cell, and just as our body, we have different organs like hearts, livers, brains, etc. The cell has different organelles, like the nucleus, or like the Golgi apparatus, or the endoplasmic reticulum. There's a mitochondria, and in the mitochondria, you can see that there's the mitochondrial DNA. Here's a bigger picture showing that it's 16,000 base pairs long, with about 37 different genes in the mitochondrial genome. And so this here is a multiple alignment. These are for human individuals. And so as you can see, that here's a sequence, and you can see that at certain positions, there are differences. There's T's and A's and G's, but there are also large swaths of sequence similarity. We have GTT, TAT, GTAG, which is pretty invariant in this mitochondrial study of primates. And so if we take 2 species, we can simply compare the DNA sequence and see how many positions they are similar at. And so we can calculate a mitochondrial DNA sequence similarity value. And so in primates, what I took all these sequences, about 30 different species. They found we have Homo, which is humankind. And we found Homo sapiens neanderthalensis. We found Homo sapiens altai, which is Denisovan. We found Homo heidelbergensis, which is a fossil human. Very interesting that they were able to isolate DNA from these fossils. Svante Pääbo, he studied Neanderthals, the Neanderthal genome. We actually took Svante Pääbo's DNA data. His group actually sequenced Neanderthal and Denisovan. They sequenced these two archaic humans. And our group at UNMC in Nebraska Medical Center, we assembled their DNA puzzle pieces into the whole genome sequence. That's what we did in our group. And so here we have also of Pan Paniscus, we have the two chimpanzees, we have gorilla, separate group, we have Pongo, and then we have Hylobatus, then we have macaques, we have 6 different primate groups, we have lemurs as an outgroup. And so I think that they isolated this empty DNA from fossils. We can still tell that Homo heidelbergensis is human based on its high similarity with other humans based on its mitochondrial genome sequence. And so again, the sequence similarity between chimps and humans, here are the two chimps, here are humans, it's a dark orangish, but the sequence similarity is only 90%, not 99%, as evolutionists purport. And also it's very interesting that with before this DNA data from Homo heidelbergensis, creationists hypothesized based on morphology alone that heidelbergensis is human based on their bone shape, based on physical traits. And now the molecular results also reinforces the same result. So here, morphological baraminology and molecular baraminology are a nice agreement with one another. It's a very outstanding result. I also studied pinnipeds. We have 3 families, Otaridae, Odobenidae, Phocidae, and Muscelidae. It's an outgroup. Found that seals, Phocidae, are one Brahmin. Walrus is its one single species in its own Brahmin. And we have fur seals and sea lions in a 3rd pinniped Brahmin. So as I mentioned yesterday, pinnipeds are an apobrahmin. They do look similar to one another, but when we look at their DNA, we're able to discern 3 putative pinniped holobrahmins. We have the seals, walruses, and sea lions. These are the 3 groups based on this analysis. I also looked at cephalopods. We have several squid groups. We have Octopodidae. We have octopuses. Octopuses are a holobrahmin with a very highly characteristic morphology. I also found cuttlefish and also Nautilus and Argonaut. Here's Daniuria, it's a fish species as an outlier. And so I discovered these 7 putative holobrahmins amongst cephalopods. Yes.
Can you define holobrahmin again?
So holobrahmin, a holobrahmin, there's the word holos and brahmin.
Yeah.
As it means the whole Brahmin. A holobrahmin is the complete exhaustive list of species in a created kind. It's like the full list, the full roster of all the species that exist in a holobrahmin. It's the whole Brahmin. It's a holobrahmin. Yeah.
Thank you.
Okay, you're welcome. So these color bars, they each represent an individual holobrahmin. Now, of course, these colors designate different holobrahmins, but of course, we might not have discovered all existing species in a holobrahmin. It's just that with molecular methods, we have to delineate and separate holobrahmins from one another. And again, like I said, that in the future, we might be able to breed new species that will belong to the same holobrahmin. Now, another study that I did, my most recent publication or study, is the molecular Brahminic placement of Homo heidelbergensis. We saw results that it is human. Homo heidelbergensis has been classified as an archaic human according to morphology studies. It has erectine jaws, teeth, body size, and locomotion, meaning that it shares traits with Homo erectus fossils. Homo erectus are also humans. has a cranial capacity, the cowboy 1 fossil, of 1,325 cubic centimeters, which is in the lower range of modern humans. But also very interestingly, that besides the mitochondrial DNA, it also has nuclear DNA as well. Neanderthals, they isolated nuclear DNA for Neanderthal. They also found a small amount of nuclear DNA for Homo heidelbergensis as well. And so what I did was I compared this DNA data that they had for heidelbergensis with a known archaic so-called Paleolithic human. And so the idea was that I compared the Homo heidelbergensis DNA fragments, aligned them to the human genome and also the chimp genome, and also reads from archaic Paleolithic human also to the human genome and also to the chimp genome. And so I calculated a variant density ratio, and it's a bit technical, so it's not a problem if you don't understand, but basically if the more substitutions there are between two sequences, the larger the genetic difference, meaning two different Brahmins. But if there are less mutations, if there's a smaller difference, it means that these two sequences come from the same Brahmin. So if you have a lot of mutations which separate two DNA sequences, it means that the average distance between two mutations is very short. This is a case where the two sequences where they come from different Brahmins. And so again, variant density is inversely proportionate to the average length between two mutations in the DNA. If we compare chimpanzees with humans, we have a lot of mutations between the two genomes. Whereas if we compare to human sequences, then there are fewer mutations, and the average length between two mutations is larger. So these are two different Brahmins, these are the same Brahmin. And so variant densities were very similar between Homo heidelbergensis and Paleolithic man when mapping to human and chimp genomes, meaning that when we look at these different, these two methods of matching these DNA sequencing reads, the pattern is very highly similar between Homo heidelbergensis and archaic Paleolithic man, meaning that Homo heidelbergensis is really another archaic human individual. So moving forward, besides the mitochondrial DNA, they also have a lot of chloroplast genome sequences at NCBI. The chloroplast is another cell organelle in plants. It's responsible for photosynthesis, capturing energy from the sun, using it to make sugars. And there's a chloroplast. Here's its internal anatomy. And in these membranes, in these thylakoid membranes, we have those enzymes which are responsible for using solar energy to transform it into energy that the cell can use to produce sugars. And so in the chloroplast, we have the chloroplast genome. It's about 10 times the size of mitochondrial DNA. We can also use chloroplast for analysis in baraminology studies. It's a bit harder to use than mitochondrial genomes simply because of its size. I did a study in liliales, which are lily species. And so here's another heat map for different liliale species. We can see that there are, again, several groups, several Brahmins within this larger taxonomical group of liliales. We have Paris and Trillium, Beatrium, Amena and Tulipa, tulips. We have lilies, Lilium, and then we have some smaller groups and individual species. So using chloroplast genome sequences, we can find different putative groups within liliales. I think liliales, it's on the level of order. So we have several Brahmins within this order of plant species. We can also look at organelle DNA. I looked at this hypothesis that within a kind, the mitochondrial genome, the following mitochondrial genome factors are fairly invariable. The gene inventory is the same. They have the same 37 gene sequences. The gene order is also preserved in within the same Brahmin. The genome is also the same length, also pretty much the same length, same genome size. And so we should be able to tell the difference between two different Brahmins based on these mitochondrial DNA characteristics. So on this figure here, what we can see is that each one of these bars, they represent one species. This bar represents the mitochondrial genome of a species. And these different colored segments, they correspond to different genes. Here's a list of the genes with the corresponding colors. So based on the color, you can tell the order of the genes in the mitochondrial genome. So in the cluster one, we have nautiluses. We have 3 nautilus species at the top. Then we have 2 argonauts. These are 2 separate Brahmins because their genome length is fairly similar. The order of the genes is also largely similar. It's also, the gene order is very different from that of other Brahmins. Here we have octopuses. Then we have 3 squid groups. Then we also have cuttlefish at the bottom, and here's Daniaria, which is an outlier species. So based on the genome length, the DNA similarity, and also the gene order of the genes in the mitochondrial genome, we find we have several different groups within cephalopods. Cephalopods, meaning it's a group of mollusks, such as cuttlefish, octopuses, and squids. So molecular baraminology methods, there are several advantages. There's a ton of data in the databases from NCBI, SwissProt, or other different databases. Mitochondrial DNA genomes are also easy to isolate and quick to sequence. We can also study microorganisms. We cannot really study the morphology of bacteria or viruses. It's also not sensitive to convergent morphology. If we remember the shark, the ichthyosaur, and also the dolphin, we can tell that these 3 species, they belong to different kinds. There are some disadvantages to these methods. If we have missing hypothetical or low quality proteins, there's also missing molecular baraminology is also plagued by missing data. Genomes might also suffer from low coverage if they're not sequenced well enough. There might be missing or ambiguous sequences, missing DNA letters. And also the mitochondrial DNA sequence is only a very short sequence. It's not as representative as the whole genome sequence. And then what is the future of molecular baraminology? Since we have lots and lots of data in the databases, we can, more and more species studies can be made possible. We can do all sorts of baraminology studies with this ton of data that we have. Until now, only 1 or 2 chloroplast genome analyses have been done. And there have been a lot of mitochondrial studies, but we can also examine chloroplast genomes, and we can study plant molecular baraminology. A gene content method has to be fine-tuned. We can do some fine-tuning here, parameterizing the different protein comparison algorithms, what counts as a protein ortholog. This might be a bit too technical, but there's a lot of work to be done in molecular baraminology. We have some algorithms that we can make use of, but we still need to fine-tune them so that we can have more precise predictions of what kind of hologramins there are using these molecular methods. And so that's it for this talk, and thank you very much for your attention. And if you have any questions, then please feel free to ask. This was, I feel, much more technical than the previous talk, but I just want really to illustrate that as creation scientists, we can do really high-level technical science, which can match the level of evolutionary biologists. And as you can see, that a couple of these studies were actually published first in secular papers, and then a subsequent creation paper was written about the same subject. So just really to keep the faith that the data really does support the creation worldview and not evolution.
Matthew, let me begin with a question that was online, if I may. This question is there anything that shows that the coefficient, JCD, maybe we're talking about Jacquard's coefficient. Yeah. Is there anything that shows that the coefficient, what the coefficient is between each and all different species, including across holobaramins?
Could you repeat the question?
Sure.
Yeah.
Is there anywhere that shows what the coefficient is between each and all different species, including across holobaramins?
Okay. If I understand the question correctly, is there like a gold standard value which can separate species from into different or the same Brahmin, like a special cutoff? There is no such golden Jacquard coefficient value number. Rather, it's very specific to individual studies. So I admit that the method does have this kind of shortcoming that if you take any two species from the databases, if you look at the gene content, and if you calculate a Jacquard coefficient value, you cannot really tell just generally if they're in the same Brahmin or different Brahmins. It really depends on the study that you're making. Like when I made these heat maps, then I did these heat maps in R, and R uses these clustering methods. Based on the clustering method that I use, we can show, it can be shown that that different species, they cluster into different groups, that we see sharp distinctions between individual Brahmins in these heat maps. The way we detect the significance of these Brahmins is when we calculate a p-value. We look at the similarity within a group, and we compare them to values between species from that group and from all the other species in the study. So we have two sets of correlation values or two sets of Jacquard coefficient values. We just do a t-test. We can measure how statistically significantly different these two value sets are. And we can say that if it's below 5%, then we have a statistically significant Brahmin. Yeah. Okay. Yes.
Where do evolutionists get this 99% similarities between humans?
Between humans and chimp.
Let me repeat the question so those online can hear it too. So the question is, how does the evolution model get to this 99% similarity in human and chimp genome?
And we see, and then the creation is what, between 90?
It's about 90%. Others put it at 80%. And so when they sequenced the chimpanzee genome, like I said, that they assumed that humans and chimpanzees are related. So they were looking for like a very high match between chimpanzees and humans. And so they looked at those regions in which there was a high level of similarity. And so indeed, that's where you see this 98-99% similarity. But if you look at the regions where the bigger differences are, where there's no match between the human and the chimpanzee chromosome, that lowers the similarity value down to 90%. If you remember if we go back to the DNA heat map for humans, for primates, we see that here are the two chimpanzees, and we see they form one group, and here are humans. We can see that this darker orange color, it corresponds to similarity values of like 90%. So if we compare modern human and chimpanzees, the percent similarity is only 90.7% when we look at the mitochondrial DNA.
In other words, it's starting out with biases.
Yes. It's sort of cherry picking the data.
Okay.
Yes, in the back.
Yeah, and this maybe goes more back to yesterday, but when we define a baramin, right, whether that's molecularly or whether that's morphologically, right, we find lions and tigers are in the same baramin. I wasn't clear yesterday. Does that mean that God created only one, right? God didn't make lions and tigers on day 6. He made ligers that then became lions and tigers? Or was that just on the ark that there were ligers that became lions and tigers? Or how do I correspond this idea of Baraminology with what
we see today.
with what God made, or what do we say that God
Yeah,
made?
that's
The question is, how do we correspond Baraminology to what God actually made? How is this helping us find out what the original created kind was?
Right. So that's a very good question. And so as to what kind of cat species were there in the beginning during creation week, we don't exactly know. I think we can assume that their genomes were much more, complete. If we look at, let's say, sand cats, they're the smallest cat species in the world. They're sort of like at the end stage of cat speciation, sort of like poodles. Poodles are very small. They have immune diseases. Sand cats also have immune diseases, and so we only know about humans that there were 2 human individuals during creation week, Adam and Eve. It may be possible that there were 100 cats created on day 6, maybe 1,000, maybe 10,000. We know that the birds, the seas, they swarmed with fish, and that multitudes of birds, they flew across the firmament of the heavens. So it may be that God created 1,000,000 birds, or maybe 50,000 birds. We don't know the number. We don't know what kind of species there were originally. We can try to guess. And so, like I said, that the wolf may have been the archibrahmin of the dog kind. Also, when you look at pigeons, I think it's called the stone pigeon. Darwin himself writes that in the origin of species, that all pigeons, they come from the stone pigeon. And so it may be that there were more individuals in a Brahmin, and when there was the flood, God obviously he took 2 of each kind onto the ark, so that after the flood he would be able to help save the animal species through the flood by taking representatives of each kind. And so, like the flood, it's sort of like a second creation, and like Noah's like a second Adam, that just as species were differentiated after creation, so they also differentiated after the flood. As to whether they were the same, whether they looked exactly the same as in creation and also during the flood, we don't know. I think it's likely that they look different. Like, for example, tigers and lions and like ligers and liligers and peligons. We see these species today. So I think that tigers and ligers, these cat species, they would be the end product of variation within the cat kind that we see today. But what kind of cat species were there earlier? We don't really know. We just have fossils. So just like Nathaniel Jensen said that we have lots of like 75% of species went extinct. There are all sorts of very, very interesting. There's a wide variety of also extinct organisms in the past.
So I'm sorry. Does that mean that you would say that there could be more than one Baraminology, right? Because maybe there were wolves and there were jackals on day 6, and those are within the same Baraminology, but he made 2 different.
Well, whether they were wolves or jackals, I don't really know.
Yeah,
but possibly we don't know exactly the thoughts of God that he had during creation week. Adam and Eve, we know for sure, 2 humans in the beginning, but yeah, I mean, again, if we look at the humankind, we shouldn't practice the arrogance of modernity that we define all of humanity based on modern humans alone. Like, because Homo erectus, Neanderthals, Denisovan, Homo heidelbergensis, these archaic humans, they're also humankind. So Noah could have looked different from us. Adam could have also looked different from us, like slightly different, but we could recognize that he was human.
Can I
Yes.
repeat that question? Because it's a very, excuse me, it's a very insightful question and comment there. What's being asked is, was the original, were there just two, was there a mating pair originally created by God, and that is the arc Baramian type, or were there multiple within that? So yeah, that's a very good question.
Yeah.
Certainly we know that at that bottleneck, at that funnel of the arc, there were for most of them just two. So that would be the archetype of the modern things.
Yeah. So the archetype, I think, very likely does not coincide with the created kind. There's no differences between the two. So
Right.
yes, you had a question.
Or a biblical taxonomy, it seems that 1 Corinthians 15, verse 39 is really foundational. That's where it says all flesh is not the same flesh, but there is one kind of flesh of men, another flesh of beasts, another of fishes, and another of birds. And I have so many questions to ask you, but I gotta try to think of this one that, tries to connect up what you're saying with that.
Yeah.
So to me, it was really striking that it does talk about flesh of fishes and another of birds, and they were both created on the same day. That's kind of interesting. But what I was wondering is how any of these algorithms that are really created by man, how they incorporate just this for starters, that there's a different flesh and between, in this case, fish and birds, and if any of these genome investigations, I mean, they seem to not even consider this. It's as if all the molecules, genes, everything boils down to kind of the same evolutionary, same kind of stuff, when the Bible teaches that there is actually very different flesh. And what does the Bible mean by flesh? Is it the molecular structure, the DNA, or what?
The question is relating 1 Corinthians 15, different flesh, and how will Matthew respond to that with his studies?
Okay, so the verse that you brought is very, very interesting. It's very useful. And if we look at the text, it mentions beasts, fish, and birds. And this is it's a Hebrew, it's not Hebrew, but it's a merism, meaning that we have birds of the air. That's one environment. We have beasts of the field, which is a dry land. We have fish of the sea, which is the marine aquatic realm. So I would interpret this verse as saying that there are different kinds of animals in the different living spheres. We have aerial creatures. We have, the Hebrew word for flying creatures is off. And so the flying creatures, such as birds and bats, were created on day 5. So here I think what this verse is mentioning is that the animals that we see in the sea, in the air, and on the dry land, they are different flesh. They're different kinds. Humans are also a separate flesh, a separate kind. They're not related to fish or animals or whatever. So we don't have an inner fish, as that evolution said yesterday. So we can forget that idea. So the verse which you brought is very useful for baraminology. It just reinforces what Genesis 1 says, that these different groups, these different kinds, they reproduce each according to their kind. Like plants, there are herbs, there are trees, there are bushes. There are really different kinds of plant species which multiply within their own kind. And so really my 2 talks, of course, we have these algorithms, we do all these studies, but we're finite. I have to stress that as a scientist, as a creationist, we hold to the Bible very strictly, but we hold to these algorithms very loosely. These algorithms might be incorrect. So we hold to sola scriptura, that these are just my ideas. I'm trying to, in my own finite sinful way. I'm trying to understand what kind of kinds God created during creation week. And so these might have to be revised. I mean, we're actually in the process of revising the gene content method to make it give more precise results. But so basically, when we look at morphology, when we look at genes, we're trying to find sharp distinctions between kinds. That kind resembles kind, and it's visually sharply distinct from other kinds. Because when Adam when he named the animals, God brought all the animals into, as a good taxonomist, since everybody is a theologian and everybody is a scientist, Adam, with his level of scientific knowledge, he was able to tell the difference between bears and horses and bovids and camels and different kinds of animals. And so it's really the name of the game in Baraminology is whatever data that you're using, we try to find statistical, mathematically significant differences between kinds. Yes.
About 40 years ago, about the time you were born, I remember reading an article in Scientific American about a chemical reaction where they reproduce chemicals, so that's where I think they started this analyzing, being able to analyze DNA, because whatever gene is, it multiplies so many of them, so they end up with the ability to extract them in the laboratory. And my question, I guess, is how many levels of technology is there between what we actually see? And the question arises, in my mind, when you talk about getting DNA from these fossils, like mitochondrial DNA is part of the cell. A fossil, how does it have a cell when it's all ossified? It's all made into stone. Where do you get mitochondrial DNA from a fossil?
Okay.
The question deals with the second part, at least. How do I get DNA from fossils? And the first part dealt with some of the technical aspects of actually extracting.
Okay. So as a bioinformatician, I worked only very, very little in a lab. So I sit in front of a computer and I write programs all day long. But I can say a few things about how they isolated DNA from Neanderthal bones. So basically, when you do DNA extraction, let's say RNA extraction, you have to take your samples, you have to break down the cells, you have to break down, let's say, these cell junctions which exist between cells. You have to break apart the cell wall. You have to use all these lytic enzymes, laser screens that you break something down. You have to use these enzymes which break down all these components. Then you have to, they use all sorts of chemicals to like really break away the cell membrane, all the other cell organelles, and then break apart, let's say, the nuclear envelope. And then they can use all sorts of buffers to contain the DNA in a buffer-like solution. But as to Neanderthal, Svante Pääbo wrote a book on how he did this whole process. Now, first of all, it has to be very sterile conditions. So like when they're doing the isolation, they have to have like a hazmat suit. They have to go through multiple doors into a special room where there's vacuum. If they breathe into the sample, their skin cells, they breathe their own modern human DNA into the sample, and it contaminates. Contamination is a big, huge problem. And so, but you see, the thing is that the fossils are young. They're not millions of years old. So, they've also shown DNA and red blood cells and soft tissue in dinosaur bones. So organic tissues are quite common in dinosaur bones and in these archaic fossils. So there can still be like remnants either of the DNA or possibly osteocytes, like bone cells in these bone tissues. And they just have to like very, very careful as to how they treat these bones, because some of these fossils, you touch them and they get powderized instantly. So you have to handle these fossils with extreme care. Now they've shown that when they isolate these DNA fragments, they get these DNA read sequences. Now, generally, when I work with read sequences, these sequencing reads are about 150 or 200 or 300 base pairs long. But in archaic DNA, they may be 35 base pairs long or 50 maximum, like very fragmented, since they have been broken down ever since, the Ice Age. Whenever the Neanderthal died, there's been decomposition. It's out in the wild. And so these DNA fragments are very short, but still they're able to sequence these short DNA fragments. And then what our goal is that if we have these DNA fragments, these short pieces, or like jigsaw puzzles. Svante Pääbo's group, he sequenced these short DNA sequence reads. And what we did at the University of Nebraska is we actually assembled a jigsaw puzzle. They provided the puzzle pieces, we assembled the jigsaw puzzle. Basically, that's what we did. And basically, that's how they isolate DNA sequences from organic samples. But then again, I'm a programmer. I don't work in the lab, so this is what I can say.